This video demonstrates an approved workflow of the Playbook Workflow Builder. Use Case 13 was designed to identify cell surface drug targets for individual cancer patients and then provide knowledge about these targets from multiple Common Fund programs. This workflow is created in collaboration with all participating Common Fund data coordination centers of Playbook Workflow Builder Partnership, leveraging several APIs from the different programs. The workflow starts with RNA-seq data collected from the profiling of a tumor extracted from a patient. This data was provided by Kids First Common Fund Data Coordination Center. The workflow first identifies genes that are highly expressed in the tumor and lowly expressed across all the normal tissues profiled by the GTEx Common Fund program. From this result, users can select individual genes to further investigate. Using the selected target gene, the workflow will return knowledge about the gene from multiple Common Fund programs. First, the workflow performs Link's L1000 reverse search query to find drugs, small molecules, and CRISPR knockouts that maximally up or down regulate the expression of the target. The query returns Link's L1000 chemical signatures that mimic or reverse the expression of the target. Next, the selected target is queried against the metabolomics workbench to identify associated metabolites and relevant metabolic reactions. Following that, regulatory elements related to the target are obtained from the Linked Data Hub, or LDH, developed by the ERCC Common Fund DCC. Finally, the glygen database is searched to identify relevant post-translational modifications, including glycosylation, associated with the selected target. To summarize, the workflow begins with a set of RNA-seq samples collected from a tumor of a single Kids First patient. The RNA-seq samples file is parsed into the gene count matrix. The matrix is then screened to identify genes that are significantly highly expressed in the tumor compared to normal tissues as profiled by GTEx. Users are then prompted to select a single potential target to explore in depth. Based on the user's gene selection, the workflow gathers information about the target from Link's IDG, metabolomics workbench, xRNA, and glygen, and presents that information as a report with figures and tables. With that, I'd like to acknowledge the members of the Playbook Partnership team from xRNA, glygen, Kids First, Links, and Metabolomics Workbench for all of their work over the past year. I'd also like to thank the CFDE community and our NIH program officers for their continued support. Thank you all for your attention.